Welcome everyone to WeScience. At WeScience, our goal is to promote local science on a global scale. Today we have a special guest, Professor Rakesh Veedu. Professor Rakesh Veedu is a scientist, is an innovator as well as entrepreneur here in the city of Perth. He is a scientific leader at the Murdoch University as well as Perth Institute, and he is a co-founder of SynGenis, which is a manufacturer of RNA DNA oligos, as well as Progenic Pharmaceuticals. Thank you so much, Rakesh, for accepting our invite to be part of this podcast. We are going to discuss your journey as well as your science and your vision for the future.
Absolutely. Thank you so much, Carlos, for the opportunity. Indeed, I'm delighted to be with you today.
Likewise.
Sure. I'm glad to answer some of your questions and also to share my journey and experience in the field of RNA drug development.
Fantastic. So before we start with science, I wanted to go a little bit in the early memories, and your motivation to becoming a scientist. So which one were your first memories of wanting to become a scientist?
That started way back. I mean, in India, during my undergraduate degree, it was a triple major course focused on chemistry and microbiology and zoology. The very first day, I can remember one of my class professor came and asked, okay, who would you like to be? And then literally to everybody in the class, right? So I was literally asking that question to every one of my friends and classmates. And I mentioned to the professor that I would like to be a person developing drugs or vaccines in the future.
Wow.
And I don't know whether it's in a good way or bad way. So I was one of the persons who said that. And then I really liked chemistry a lot. So I wanted to be in the field of drugs and drug development. And that's how I started in my early days.
And what was your motivation of drug development, etc.?
It was kind of, through reading a lot of newspapers and also activities around the pharma sector, making drugs and manufacturing drug materials or drug product in India at that time. And I didn't even know how they are made and what are the stories or science behind it. I had no clue at all. But I thought maybe I should do something in that area moving forward, in my future career. And that's how I embarked on and chemistry always I liked a lot.
Wow, that's a fantastic story. Now you are in the drug development area. So
yeah, it's a coincidence.
Well, it's part of your dream. And then you decided to come to Australia. Why motivated you to come to Australia?
Yeah, I came to Australia about 25 years ago, mainly to pursue my master's degree.
Yep.
After my undergrad degree in India, I wanted to do my master degree. And my father basically sent me to Australia to do my master course. So the idea was actually to complete my higher education in Australia and go back to India and pursue my career in India, whatever you can possibly get. And that was the whole idea of sending me to Australia by my parents. And I said, okay, I'll do it. But after coming to Australia, completely changed, right? So then I didn't know that, if I stay on, I have better opportunity here in terms of the field that I really wanted to pursue. And that's how I decided to stay on. And it's purely because I also found positions at the universities. As a part-time opportunity, I was lucky enough to be employed at Griffith University's AstraZeneca laboratory.
Oh, wow.
R&D facility as a lab assistant at that time. And, I've been working there nearly about a year while I was doing my master's. And I used to talk to my senior professors and also the research people. They all have PhDs and higher degrees, and I wanted to pursue my PhD afterwards. If I go back to India, probably I might not be able to do any of those. So I thought of, staying back in Australia, continuously talk to people on how I can pursue it. So luckily I was able to, after completing that master degree, and also that year time at AstraZeneca, I was lucky enough to be, at the University of Queensland to do my initially MPhil and then later the PhD. And that's how I kept going.
And then you did PhD at
I ended up doing a PhD at the University of Queensland under my mentor, Professor Kurt Wenthrop. Yeah. So,
and how the journey? So what were the lessons, failures, and the success of those times? Because it's early times in PhDs. It's very challenging.
Indeed.
And it can be very challenging. So what keep you motivated to go through that journey?
Yeah. Well, for me, it was just one after the other. I would say that doing PhD at that time wasn't an easy thing to do. Obviously, you had to have scholarship to do a PhD. So initially, I wasn't able to get scholarship. And then my Professor Wenthrop, managed to give me like tutoring to support my living expenses. Obviously, there was no tuition fee at that time. And then I was living with very tight budget. That was still okay, but my passion is to continue in chemistry.
As a migrant doing
as a migrant doing PhD for a degree. And my Professor Wenthrop, wholeheartedly supporting me to do my PhD. And I used to work very hard to create a lot of, antibiotic compounds like quinolones and naltharidons. I can even remember me leaving the lab at around 12 p.m. Around 11:30 p.m. in the night. I used to do a lot of column chromatographies to isolate those compounds, after, flash vacuum pyrolysis and followed by nucleophilic addition reactions and so on. So many columns in front of my bench, and I set up. And while I complete all of those color chromatographies, it's about 11:30 p.m. So we used to work hard. And luckily, about 6 months later, my professor found full scholarship for me. And so it was a tough journey of doing PhD at that time. And then, in 2005, December, I managed to submit my PhD thesis and leave for Denmark for postdoc.
Oh, wow. So tell us, please, a little bit more about the Denmark experience as a postdoc.
Yeah, that's how I changed my hardcore synthetic organic chemistry to a bioorganic chemistry, in the sense chemistry related to RNA and DNA synthesis, manufacturing, and drug development. It all happened at my postdoctoral time with Professor Jesper Wengel at the University of South Denmark as part of Nucleic Acid Center. So I was lucky enough to be employed as a Danish National Research Foundation postdoctoral fellow at the University of South Denmark for about 4.5 years. And I was working with Professor Jesper Wengel, as I mentioned. And my mission was actually to focus on developing LNA-based therapeutic molecules or aptamers and targeting ligands and so on. That's my postdoctoral time and career. So that's where I changed my field to RNA therapeutics and RNA drug development industry. So I was so lucky to have such a pioneering person like Jesper Wengel as my mentor and supervisor for postdoctoral period. So I learned a lot from Professor Wengel.
He is a global authority on
LNA chemistry, and also I learned the basics of all about RNA drug development, antisense, aptamers, and about the modified nucleic acids. And he had an exceptionally talented bunch of researchers with him from all parts of the world, actually. So it was a very good time in the industry. That RNA therapeutic was not as matured as the way we currently have. It was all in its infancy. Only a few companies, Ionis Pharmaceuticals, if I could remember, Ionis and Sarepta and Alnylam, and these were the few companies around at that time. But it was a great time as part of my career, and also a person who wanted to learn more about this, the art of drug development. So I learned a lot from Professor Wengel's group, and again, I'm blessed to have a mentor like Professor Wengel.
Yeah, my next question is, how was his mentorship? Because he is an authority, right? And he is a scientist, but also innovator. He has many patents, as well as he has co-founded many different biotech companies and pharma. Yes. So how that, and yourself is also, the similar profile. So how his mentorship helped you during your postdoc?
Oh, he did a lot to me. Professor Wengel, if a person work with Professor Wengel, they all obviously will admire the way Professor Wengel's way of supervising people, and also helping us to gain more knowledge, and also to help us achieve our milestones in terms of projects and in terms of knowledge base. Yeah, he's very, very supportive. And I'm, again, very fortunate enough to be associated with Professor Wengel. And as you mentioned, that he obviously made significant contributions in our industry with respect to the modified nucleic acids. For example, locked nucleic acids, obviously co-invented by Professor Wengel. And he obviously working closely, and he successfully translated that to industrial commercial development. And two companies came out of that single molecule called Exicon, was focusing mainly on the manufacturing side, and Centaurus Pharma focusing on developing RNA-based therapeutics. So yeah, I mean, as a young person like me, witnessing all of those together with the scientists who developed all of this is absolutely rewarding, and also a significant learning curve. Yeah. I mean, I'm very thankful to Professor Wengel.
And it must be very exciting times.
Yeah, that's right.
Yeah. So then you decided to move to Australia.
Yeah, after nearly about 4.5 years of my postdoctoral period with Professor Wengel, I decided to get back to Australia. It was also mainly because of my family life. My wife wanted to get back to Australia, and I was lucky enough to get the UQ fellowship and then get my independent career started at the University of Queensland within the School of Chemistry and Molecular Biosciences. And I was at this Molecular Biosciences building 72. And I started my career, independent career, in June 2010. And I was, again, lucky to have two mentors, Professor Rose Smith and Professor Joe Rothnagel. I think Alan McEwen was actually the head of school at that time. It was a very good journey.
And then you were recruited by Murdo University to establish your own group.
Yeah, after 3 years of my research at the University of Queensland, wherein I focused on developing nucleic acid-based therapeutics, mainly for breast cancer and Alzheimer's disease. And throughout my stay, we managed to come up with a lead molecule for breast cancer called RNV66. And after my time at the Uni of Queensland, we moved to the Murdoch University here. Yeah, so it was recruited by Parent Institute, but I was based at the Murdoch University campus. So that's how I ended up here.
Academic life as a scientist is very busy, but it's very rare to find someone that's able to fund one biotech company. But then you decided to found two companies. Yeah. So one biotech, which is SynGenis, and the other one is pharma, which is Progenic Pharmaceuticals. Yes. So what was the motivation to do that, to be able to co-found those companies?
Yeah, well, that's a great question. So I've been, running my academic laboratory at the university campus for nearly 6-7 years. I mean, I started my career here in 2015, right? And around the time of COVID-19, we had, I hope you remember, we had a strict border closure in March 2020. Nothing was coming in the country. I mean, we were fully restricted.
Totally blocked, yes.
Yeah, totally blocked. And that time, the entire scientific community realized we were so reliant on overseas companies to, get all of our oligonucleotide supply, right? And IDT, integrated DNA technology, almost exclusively monopolized that as an overseas supplier for entire Australia. And I was contacted by Department of Industry, federal government, to manufacture the RT-PCR primer. And at that time, I was only manufacturing that at the laboratory scale. And I was one of the labs in Australia capable of manufacturing DNA and RNA oligonucleotide. But I could only manufacture just enough for a lab and also my collaborators around Australia, but not for the entire country as a supply chain, making grams of such material. But that really kept a seed in my head now is the time to do something. I wanted to convert that into a commercial operation so that the Australia and New Zealand can benefit out of what we do and what we're good at doing at a laboratory level or an academic level. And I immediately contacted my mentor, Professor Marvin Karatas at the University of Colorado, and also at my institutional heads, Steve Arnott at the Parent Institute, and also our Deputy Vice Chancellor of Research at Murdoch University, and also my mentor at the university, Professor Steve Woolton. They all wholeheartedly supported the idea of converting that lab into a viable business, in the sense to support our ongoing activities at the universities and also the whole academics and industrial sector in Australia. I worked a lot on developing a plan, how to execute that since end of March 2020. And in the same year, December 2020, registered the company SynGenis. So that's how we started SynGenis, mainly as a manufacturing organization to supply RNA and DNA oligonucleotides in this country. So I became, I would say, a forced entrepreneur, and that's how I put the activity around that. And 2 years later, obviously, I've been developing a lot of therapeutic molecule based on RNA for rare and also acquired diseases, mainly for type 2 diabetes at the university for a long time, together with my mentor at the University of Colorado, Professor Marvin Karatas. And after nearly about 6 years of our research, or 6 years of our research, we came to a point that it needs to be converted into a commercial development. And Marvin and I formed ProGenis Pharmaceuticals, mainly to develop the lead molecule that we have identified at the university and Parent Institute, to develop that, commercially. And that's how ProGenis Pharmaceutical born in the end of 2022. And we began our operation in full-fledged, in full swing in early 2023 at Bentley Technology Park in Bentley, Perth. So that's how we started with ProGenis Pharmaceuticals.
Wow.
So now the company ProGenis is going very strong. So our objective is basically to take our lead drug candidate to first in human clinical trial by the end of 2026.
You mentioned your mentor, Professor Marvin Karatas. He is a pioneer in the synthesis of oligonucleotides.
Sure.
What is his mentorship means to you?
Yeah, well, that means a lot. Marvin and I obviously came together at a conference, while we were discussing around the same chemistry. I was making one of the variants of the chemistry that we currently pioneer. And then obviously Marv had different idea. So I was presenting the biological side of the chemistry that I was doing, and then we somehow started our collaboration to mainly to explore Marv's chemistry and also the biology side, what I was planning, I was doing at that time. And that's how we were doing that thiomorph chemistry as a platform for RNA development. It is a great experience for me to be associated with Professor Marv Karatas. He's, as you know, is a pioneer in oligonucleotide manufacturing sector. So Marv obviously revolutionized the industry of RNA therapeutics, I would say, or oligonucleotide industry. So because of Marv's DNA synthesis chemistry, all of these manufacturing and companies do exist at the moment. So it is a paradigm shift in 1980s, I would say, the way we manufacture oligonucleotides now of our DNA. So with that, Marv and his colleague established a company called Applied Biosystem, which you probably know. And later, Marv also co-founded Amgen Pharmaceutical as well. So all around this core technology, it is a wonderful experience for me to be associated with Professor Marvin Karathus and also to learn every single day. And I still continue to learn from Marv from both the sectors, from the business sector as well as the academic side as well. So I work closely with Professor Marv Karathus, and I'm very fortunate enough to be with a pioneer, a global leader like Marv.
Yeah, that's fantastic. The mentorship is so important for the success of
in our career, it's really important because if we know 10%, the next 5% I could actually, or even more, I could gain. At least I could get strategic directions and what are the potential options that I might have ahead of whatever I'm trying to develop or embarking to. So that's why mentorship is key. And you always want to make sure, work with pioneers who have been through that journey and try to learn from them and also try and, do good stuff that ultimately will change the world or benefit someone in the industry or maybe requiring a key drug for treating a disease at the end of the day.
Fantastic. And I'm going to change a little bit the gears in the discussion. And thank you for sharing your journey. We're going to jump into science. Before COVID, as a scientist, if we say like a RNA, the word RNA, people will look at us like we are crazy. What is RNA?
Right.
But then during COVID and after COVID, RNA became, like a lot of development because of the RNA vaccines for COVID. RNA therapeutics or RNA drugs is evolving rapidly. So that at the moment they are for treatment of rare muscular, genetic, metabolic, infectious, and ocular conditions. And there is also a lot of RNA drugs that are in the early phases and also late phases of clinical trials and preclinical testing as well. So could you please explain what is RNA therapeutics? What does RNA drugs mean?
Right. Yeah. We are very familiar with small molecule drugs, chemical drugs, chemical molecule. We always take that. A simple example, paracetamol, Panadol. When we have flu or anything, we simply go and take Panadol, right?
Yeah.
So it's just a paracetamol. So it's basically a small molecule drug. So traditional small molecule drugs target the protein or the pathology directly, preventing the further mechanism of action. But RNA drugs, we can target the gene transcript at a very early stage to control or regulate the expression of these causes of diseases at a very early stage. So that's the major difference. Sometimes traditional small molecule drugs cannot actually do that job effectively like the way RNA drugs can do the job. So the central dogma of this is actually we have our genetic information is stored in the form of DNA. It's a long, twisty, curly molecule, right? And the information is then passed through genetic messages, which is what we call RNA.
Yep.
It simply carries the message of this information and then translate into proteins for various functions of our daily life, right? So sometimes these pathways can be regulated at a very early stage. For example, it's a very complex process. Sometimes there can be mutations, so there can be spelling mistakes or errors in this process at the gene level, gene transcript level, which will then produce corrupted messages, right? Which will then make bad proteins that are either not expressed or underexpressed, or sometimes they are overexpressed. So then we have to control or regulate the expression of these bad proteins or these proteins that are implicated in the pathology of a disease. So RNA can effectively do that. So we basically take an RNA molecule or RNA-like molecule to target the defective area or the specific area of our gene transcript to do its job and to control, that can then control or regulate the expression of these disease-causing proteins. That is the fundamental difference with a small molecule drug versus an RNA drug. And that's the impact of our technology versus traditional approaches.
Yeah, and also because when the message is transcribed and translated into a protein, then the small inhibitors, the small drugs, you had to play around all the combinations
Yeah.
to inhibit a particular protein, because all the different changes in the conformations of those, the matching between the proteins, the pathogenic proteins or the disease-causing proteins, as well as the small inhibitors. Correct. To be able to block the particular protein. So then RNA facilitates that. Absolutely. Because it's basically controlling the expression of a particular gene that's pathogenic before it's transformed into protein.
Yeah. So especially if you have multiple variants of the same proteins, so if it shares the same homology between these proteins, it might be harder to target the actual protein that is implicated in the disease. But using RNA approach, you can precisely control at the gene level on what you are targeting and what are you trying to achieve at the protein level. So that's also one advantage of RNA drug.
One of the things that I like when I joined Progenic, I started delving into the RNA therapeutics, is that my background is in cancer. So basically, in cancer, there are particular genes that you cannot target by small inhibitors because they are deemed as undruggable. So what RNA therapeutics brings to the table in terms of cancer treatment is be able to treat those particular genes that for conventional therapy is undruggables, but we can control the expression of those particular genes by using the RNA drugs.
Definitely. Yeah, that's what we do at ProGenis Pharmaceutical on a daily basis. Mm-hmm. So we develop RNA therapeutics for a range of different indications. And nowadays, RNA is not alien for people because everybody during COVID time have done RT-PCR. Okay, RT-PCR. So everybody knows about RNA, and everybody got a vaccine called mRNA vaccine. So the word RNA is not unusual, or is not a word that is alien for people. So it is still recognizable.
So you mentioned about the RNA drugs, and we're discussing about that, but there are different types of RNA drugs. Could you please explain to us what the different types of RNAs?
Yeah, sure. There are different types of RNA drugs. In RNA is a newer class of drugs. So we have antisense oligonucleotide. Again, they target messenger RNA and impose a steric bulkiness to control or regulate the expression of the actual protein. And we also have siRNA, which is small interfering RNA. These are short pieces of RNA of about 21 nucleotide in length, which are double-stranded, and which is actually targeting, again, messenger RNA in the cytoplasm, and to target and cleave the target mRNA, which is the RNA responsible for making a specific protein. And we have also microRNA, and these are in two ways. You can actually use microRNAs to overexpress some proteins which are not expressed, or you can also use anti-microRNA, which is antimer or antagomer, which are to target the microRNA which are implicated in the pathology of certain diseases, completely inhibit the mechanism of action. Mm-hmm. And then we also have other classes of drugs called, recent advancements are, CRISPR-Cas is another. So basically you can edit the gene at the gene level and control, regulate the expression of your disease-causing gene. That is more like a permanent fix. Mm-hmm. But the conventional RNA drugs like antisense, siRNA, microRNA, or DNA enzyme, so these are, they're not permanent, but we still need to, it depends on, maybe you can probably get away with once in 3 months dose or once in a month dose like that. Mm-hmm. But with gene editing using the CRISPR-Cas technology can last forever. Yeah. That's one option.
That's more permanent.
That's more permanent, I would say.
And also mRNA.
Yeah, mRNA is actually a vaccine approach, and so that's basically these are all synthetic, and the other one, the mRNA, is a kind of a vaccine that we have all used during our COVID period. So mRNAs are actually a long RNA molecule, biologically produced by in vitro transcriptions, And then that can be used also to, upregulate the proteins that are missing, and also it can be developed as a vaccine.
And also as a gene replacement.
As a gene replacement.
Particularly in cancer, for example, when there is a gene that is mutated and there are different mutations in the particular gene, you can replace with a wild type or normal copy of the mRNA for the particular
gene. That's basically
gene.
what I said about the RNA as a drug molecule and RNA as a vaccine candidate as well.
What will be the characteristics to pick particular one or the other?
Well, it's hard to answer that. I mean, what I would say is they all are wonderful technologies. And in certain cases, you may want to use antisense technology. If you want to, splice switching, for example, if you want to edit RNA splicing, then you may want to definitely use antisense drug. There is no way you can use siRNA for that. So in certain cases, obviously, we are better off using siRNA because that technology is far more potent compared with antisense, I would say. And in the vaccine scenario, you definitely, if you really want protein upregulation or the transcript replacement, then you have to use mRNA technology. And if you really want to edit certain part of a gene and to develop as a kind of permanent, you have to use like CRISPR-Cas. So these are, again, I'm saying all of these are brilliant technology. It's a portfolio. It's a class of different RNA therapeutics. They all have its merit. You use them as you need it. So I don't say one technology over the other. So it depends on what we are doing and what is the right approach and what is the right technology to develop or to cure that specific condition. And that's how we select. And that's what we should consider.
RNA drugs are new therapies. So what are the challenges for the translation of those RNA drugs? Because in COVID, everything was fast-tracked. And then there was a lot of discussions in the communities and among scientists about the safety, about the efficacy, etc., of those particular drugs. As an expert, what are the main challenges of the RNA drugs that scientists like you are facing at the moment?
Yeah, well, that's a very good question, I would say. Well, mRNA for COVID is a wonderful example of what we can do. And that's really a game changer for our field to reinforce the potential of RNA therapeutics. So in terms of challenges, obviously, we have to address the way we deliver the drugs to the disease-specific sites. And that's a key challenge that we continuously face. Wonderful technologies are coming through and evolving on a daily basis to address those challenges. If you want to deliver to liver nowadays, we have GalNAc technologies. Basically targets the Asia-glycoprotein receptor at the liver hepatocytes, and then deliver the drugs via receptor-mediated endocytosis inside the liver hepatocytes. So that's one came from Mano from Alnylam. It's now based on that there are about 3 or more drugs on the market already. So and more such innovations are coming for target-specific delivery of RNA drugs. And moving forward, this will evolve even better, and that delivery will also slowly be better for delivering RNA drugs. And the potential of our field of RNA therapeutics are immense, I would say. If I could give one example of what we can achieve in terms of RNA therapeutics, is like, it can be used for N of 1. Very rare disease conditions who otherwise could not be treated can now be treated using RNA therapeutics. So if I could grab one example is Melazin. Melazin is actually a drug specifically developed for one child called Mila Makovic. As far as I know, Professor Tim Yu at Boston Children's Hospital diagnosed Mila with a rare disease condition called Batten disease. It was mainly because of a mutation in CLN7 gene. And he came up with this antisense therapy. From the diagnosis to the development to the injection to the patient, all happened within about 10 months' time. So everybody had to work together, the regulatory landscape and the drug development landscape, and the diagnosis and the patient side, everybody had to work like a team to develop something like that, to administer the drug finally to benefit that child. And that's, I would say, is a wonderful demonstration on what we can do with RNA therapeutics. Now think about that in a global scenario. So we have that potential and capabilities of these technologies to deliver to the people who are desperately in need of personalized drugs, and that can be achieved with RNA drugs. So delivery, as I said, is one of the key challenges. And I would say to some extent cost is also one challenge which we have to address. And as we evolve this field, I believe this will all become a norm. Nowadays, you imagine like how many companies are popping on a daily basis in this industry, which is an absolute boosting story for investors and also for, for the patient community as a whole, because they will be the ultimate beneficiary of all of this development. So yeah, I mean, RNA is going to be a new industry, a new therapeutic class, which will be like a normal class in the coming 10 years, I would say.
Yeah, particularly the challenge is also the RNA drug to be able to target the particular cell that we are after, right? And with minimum toxicity, with minimum side effects, and be able to sort of ensure that the treatment is working.
And tissue-specific delivery, I mean, don't get me wrong, it's only for RNA drug. It's also there for small molecule conventional drugs, right? So they're also trying to address that. But it'd be great if you can achieve specifically for the RNA drug. That'll be a big boost because we obviously have 4 different tissues in our body.
And in terms of challenges in manufacturing, so what's your comment about that?
Manufacturing, currently we don't have a GMP manufacturing facility in Australia for RNA drugs. But obviously federal government and also there are private government partnership basically contributed to some sites in the East, and for which might come up maybe in 2 years' time, for basically for messenger RNA, mRNA drugs. Now, for small synthetic RNA, I would call Perth is a hub for small RNA drug development, because we have a history of translating 3 drugs on the market on RNA therapeutics. All 3 of them come from Perth. Basically, Professor Steve Wilton and the C. Fletcher basically developed those 3 drugs for the treatment of Duchenne muscular dystrophy. So we have a history. So on that's our strong point. And Murdoch University is obviously taking a lead charge, and Parent Institute is doing their best to support this ecosystem. Companies are also, companies like ProGenis Pharmaceuticals and Black Swan, PYC Therapeutics, these are all companies focused exclusively on developing small RNA drugs. And also we've been supported by a Western Australian government to establish a clinical grade RNA manufacturing facilities, small RNA manufacturing facility in Perth, which will come up maybe in about a year and a half time from now, which will obviously support the ecosystem, not only for Western Australia, for the entire Australia. So this is all evolving, and yeah, we can definitely see Australia taking a lead charge in this segment, in this industry. And don't forget that Australia is the only country, I would say, that have a dedicated RNA blueprint, which was announced last year at SynGenis by the federal ministers and the state government minister. So that's a big, the government clearly identified that this is an area that they have to prioritize and is supported very well as well.
Absolutely, yes. There is a significant support from the Australian government as well as from the state governments, be able to support not just manufacturing as well, but also preclinical facilities as well as clinical trials and commercialization.
Yes. So the point of hub, meaning it involves the drug design and manufacturing and clinical trials, all happening in Perth. So that's why I wanted to say Perth is evolving as a hub for small RNA drug development.
Yes.
So hopefully it'll come true.
Hopefully, yeah. So in clinical trials in the conventional drugs, the failure rate in clinical trials is around 80%.
More than that.
Yes. So what would be the lessons that RNA drugs they need to take to be able to sort of improve the success rate for all those RNA drugs to passing into clinical trials and then human?
Yeah, well, failures are part of our development, journey. So that's not unusual. What we can do is basically to learn from each, pipelines of development. So what are the problems encountered by a specific candidate for that specific indication? So what really happened? So we correct and rectify and develop a better design and develop a better RNA drug molecule to address those, limitations associated with the previous compound. So that's how we continuously learn and evolve this drug development art. So yeah, you are absolutely right. About 90% of the drugs might not make it all the way, which is still okay, I would say. So we have to start. I mean, we need to really take initiatives of developing RNA therapeutics. At the end of the day, why do we do all of this is basically to benefit somebody who's actually do not have any other alternative or better, or looking for better alternatives. And that's why we embark on this field. And that's my passion has come from there as well, to try and do I'm very good in doing this RNA therapeutics and RNA drug development. And that's what I, enjoy doing. So I obviously am very well aware of the landscape, and I'm constantly watching the drug development and the clinical trials and the failures and success stories and all of those. So we focus and we learn from all of those, and we focus on our development pipelines to make sure that we do our best to make sure our drug is going all the way.
Yeah. And also the support from the government is fantastic, but it takes a village to be able to bring, and also lots of money to be able to bring a drug into human. So in that aspect, what sort of are the challenges and that we are facing at the moment?
Challenge is money. So that's the straight answer. Yeah, it is a multi-million-dollar exercise. This government gives some minor portion of that. But obviously, we need to attract a lot of investment. With that investment, it might take us to certain milestones. So with the successful achievement of our initial milestones, we can go back to the investors again, say, yes, we have been able to achieve this milestone. Now we need to get to this. Would you be interested in funding our next step? So they might. So it's very difficult from, to get support for preclinical development. But if you are a clinical stage company, your chances are much more to gain or get investment from private investors or VCs. To get to that first in human trial phase is the most challenge that we face. And that's exactly what we are going through at the moment, but we are fortunate enough to be supported by investors. And Marv, being a co-founder of ProGenis Pharmaceutical, took a lead in funded the seed capital in our company. So that is an excellent incentive. So being a co-founder, you yourself is funding your company because we believe in this. And that gives an incentive for other investors to believe in you as well. So we took a major chunk of the money in the company. So we achieved certain milestones, and that gives us a good story to go back to another investor to support moving forward. And that's what we've done. And we've been able to get funding to reach our next milestone. So but still we need a lot more money. And obviously it's a multi-million-dollar exercise, and phase 1 will about 10 mil. Phase 2 will be about 50 to 80 mil. So yeah. And also we look forward for big industry partnership as well to take our drug molecules to phase 2, phase 3, and to market.
Yeah, you mentioned that Western Australia to be RNA drug.
Absolutely.
And what do you think in your vision, what are the main challenges in WA for drug development?
Yeah, these are all the challenges. To get enough support in terms of money. And also we face challenges to get the right people. The reason I say that we don't live in Boston. Boston always in the US, they have a history of having all of these successful companies most of these companies having based in Boston, and they do have very talented and trained people who know the regulatory landscape and also the preclinical and then the clinical development, and all of those are very well established there. So there's no shortage of people to find. So they keep on jumping from industry to industry to industry, but we don't have that here. But we are building now. Now is the time that we focus on creating all of those capable talents in WA. And Tiberak University and Parent Institute are doing their best to generate such talented pool of trained scientists who can actually do this. Now, we've got MTP Connect and various other Life Sciences WA and other agencies similar in other eastern state as well. They're also doing their part to make sure to identify the gap and to address the issue. So we have been fortunate enough to get the right people, but obviously we need to generate more and more of such talented pool in Western Australia. Money is one thing, and trained and capable scientists are another thing.
So as an academic, you co-founded SynGenis and ProGenis. What are the hard lessons that you learned through this journey?
Yes, it is always a learning curve for me. On a day, every day is an experience for me to take on to, do something new. It's very stressful being an academic at the university, at the same time be an entrepreneur running these two companies. So you can only do one company. So recently we found people to drive SynGenis separately. And I'm focused mainly on the ProGenis Pharmaceuticals. Yeah, so it is a learning process on a daily basis on various fronts, on the management side, scientific side, and dealing with people, and I would say, and also the hardship of getting money into the company to support the activities and also to achieve the milestones in time, and getting the data sets in time, and deal with the regulatory people. It's a full-on job. We are trying our level best actually to make sure that one of our programs are ending up in clinical stage, or at least in first in human clinical trial. So so that's what I would say regarding my experience running these.
And then what advice would you give to the Australian scientists who would like to innovate and as well as become an entrepreneur?
Yeah, first we should have a novel scientific technology which has been very well characterized or established in our laboratory, generate maximum data at the university or behind every technology, even if you have proof of concept demonstrated, especially with respect to drug development, in vivo or in the animal model, then is the time that you apply for intellectual property or patent or provisional patent application. And then to take it commercially, again, you have to reach out to potential investors yourself, reach out and work very closely with the university to get to that exposure with respect to multiple investors to invest in that technology. So it doesn't mean that we have to form the company ourselves. We can also reach out to investors and they can come up with a model, a solution to take it commercially as well. So work closely with the University Research and Innovation Office and believe strongly in our core technology. Without strong and compelling science, it's literally impossible to be successful in a commercial landscape, and it's very hard. And the science should be proven, science should be correct, and then getting money obviously is also a challenge, but it'll come as long as we have a credible reason, credible data, and a credible roadmap where we really want to be in 2 years, 3 years, and 5 years time frame. And that's what we have to generate and have an excellent business plan. And without having a compelling business plan, no one will invest in any company. And that's really important. That's, we have to, be thorough in what we are pitching and have a simplified pitch ready to investors that they can easily follow what we are talking. And these are all some tips and ideas that I've been applying, and then also maintain a positive attitude towards everything. And there may be people who may say otherwise. Believe in you, So obviously there will be people with negative mindset, positive mindsets, but these are all part of the journey. I mean, be prepared to accept all of those and do the best, what you think is best for you. And yeah, move forward.
What skills do you think that next generation of scientists should have?
Resilience.
Yes.
And I would say a core expertise in something, what you do. Without that, it's impossible. I mean, we can criticize all other people, but if we are not good in what we're doing, there's no point. I mean, we should have a strong scientific skill or a strong background in anything, at least in one segment of work that we do. That has to be our core strength. And then the remainder, obviously, we can partner or collaborate with, which we are not good at. So, yeah, so that focus on gaining a key skill set and a strong scientific background, generally, and also the positive attitude and the confidence come. Having all of these incorporated, you naturally start gaining confidence in you. That's probably what I would suggest.
So which skills do you think when hiring for a startup?
All of these collectively. Hiring for startup is basically based on the need and the skill set and the right attitude. And all of these are paramount because startups environment is totally different from a fully established company. Startup company only require what is required for the company. So we have to make sure that although we take people for one job, might not be the only job that they might need to do. So basically, so it's a mindset, the people with the right mindset to do everything and to take or to achieve the goal in the projected time frame. So we have to have that mindset to do outside the box. Yeah, it's a passion. So those passionate people are important. So it's not a 9-to-5 job. It's not a job that we want to do for paying our mortgage or something. So we need to really make sure it is a task that I take within me to accomplish as part of a team successfully, or at least give our best effort to take us or take the company's objective to where it is projected to reach.
You mentioned resilience as a core skill. Failure is a big part of the scientific process.
Definitely.
So could you please share a story of failure?
How many I can? There are so many, a lot of failures in terms of grant writing, I would say. So I've been writing grants and grants and grants and so many grants. I've never been successful. So I've been writing grants since 2010 independently for National Health and Medical Research Council. So I've never got a grant in myself. I mean, finally one of them came through together with my good colleague, Andrew Curry. Once with Lisa Chopin at the Queensland University of Technology came through that I was not the principal, the lead CIA, but I was one of the CIs, and that was in 2013, 3 years after. And then came with my good colleague, Professor Andrew Curry at Murdoch University, as a sepsis project that we did. We got nearly $1 million grant for that specific grant. And obviously Andrew did a great job there as well. So I think that's one of the failures that I consistently had. But after that, definitely since 2015, I've been fortunate enough to have multiple grants. But again, what we learned from all of those previous grant writing story and how to write a good grant. So this was actually a skill set that I developed over several years of grant writing. That's one. And also the resilience obviously is key element to address all of those. And also to keep your battle as an entrepreneur going forward on a daily basis is important. There are several setbacks that you will see. Sometimes you may experience some bad experience from people. Then you just you keep moving because you have a much bigger mission ahead of you. So these are all experiences that we take and learn from and move forward and still continuously believe in yourself and give our best. So these things will happen. There are good people, and always focus on advising you on good terms. So Rakesh, if you're doing good stuff, okay, you may want to focus on something else as well. That's a good piece of advice. Although criticism, it's good for you. So you listen. There are some people who may be overly negative. So then take it as a learning experience, move on, and focus on what you need to achieve. And that's, it's all part of the journey. Yeah, it's there. And that's what I would, say from my resilient side.
Yeah. So my last question is, what is your vision for the future of your science?
I want to basically develop a simplified way of designing better RNA drugs, and that's currently we are developing at the moment. That means a total paradigm shift on the way you develop RNA drugs now, the design RNA drugs now. So that's which might give you like 30-fold potency with respect to its efficacy. So that's something what I'm working on. And also I want to create success stories. I want to develop at least 1 or 2 candidates in human clinical trial phase through Progenic Pharmaceuticals. And I want SynGenis making clinical grade oligonucleotides, supplying such materials to people in Western Australia and entire Australia. So these are all good visions, and also I want to develop more and more newer technologies and contribute to the development of innovative technologies through Murdoch University. And these are all part of my vision. And at the end of the day, I'm heavily focused on developing Western Australia as the hub RNA, small synthetic RNA drug development. So I'm very fortunate enough to be collaborating with various good colleagues within Perth at different institutions. So I would like to continue my collaborations with all of those wonderful people, and we continue to do very good science in Australia, in Perth, in Western Australia.
Thank you so much, Rakesh, for sharing with us the inspiring stories, as well as your journey as an entrepreneur, as a scientist as well.
Sure.
So I really appreciate your time, and it's been an honor to talk about RNA therapeutics, but also about your journey. So thank you.
Thank you very much, Carlos. Thank you for your time.
Yeah.
Thank you.
And thank you everyone for joining into our podcast, WeScience. We see you next episode. Thank you. Bye.